Bismillahirrahmanirrahim. Production of blood cells by bone marrow are under control of group protein called growth inducer and growth differentiation inducer. Today we will discuss growth inducers and growth differentiation inducers. Inducers are reproduction of different stem cells are controlled by multiple protein called growth inducers. Four major group inducers have been described. One of the most important growth inducer is interleukin-3, which promote growth and reproduction of virtually all different types of chromatid stem cells. Chromatid means those colonies of cells which are chromatid for production of either white blood cell or red blood cell or platelets. Where the other inducer induce growth of only specific type of cell. Interleukin is responsible for the production of all type of cells, and the rest of the three are responsible for a specific type of cell. Either they produce red blood cell, they produce the white blood cell or the platelets, a granulocyte or agranulocyte of platelets. Now there is differentiation inducer. The growth inducer produce growth but not differentiation of the cell. For differentiation, this is a function of another set of protein called differentiation inducers. Each of these cause one type of chromatid stem cell to differentiate one or more steps towards a final adult blood cells. Like in one step, it occurs in the megakaryocyte. From megakaryocyte there is shedding of the platelets. One step in red blood cell, there are multiple steps. Also in the white blood cell synthesis, there are again multiple steps. Here in this diagram you can see the growth inducer cause production of all type of cells. Multiple cells, multi-different cells in the blood, multipotential hematopoietic stem cell or hemocytoblast, and when the growth inducer acts on this cell, it causes production of a particular type of cell. Like either it may be produce a progenitor cell for the RBC or progenitor cell for the WBC. So when it causes growth of progenitor cell for the red blood cell, that is proerythroblast, and this proerythroblast then produce the red blood cell. Or when it is myeloblast or lymphoblast progenitor, they produce lymphocyte or monocyte, or produce the granulocytes, the basophil, neutrophil, eosinophil and monocyte, and macrophages. The stem cells are capable of producing their own copies. During embryonic life, they are also capable of producing all type of tissue, including the blood cells. They are producing the nerve cell, the liver cell, the cardiac cell and the muscle cell, also intestine and kidney. These cells are actually multipotential during embryonic life, and after birth most of these cells are become silent. There are three types of these stem cells. One is the embryonic stem cell, the other is adult stem cell, and the third one is pluripotential stem cell. The embryonic stem cells supply a new cell for an embryo. These stem cells are said to be pluripotential, which means they can change into any cell in the body. Meaning they are pluripotential, they can produce bone, they can produce muscle, they can produce kidney, they can produce heart, they can produce skin, hair, tail. All the organisms develop from these cells. And adult stem cells supply new cell as an organism grow and to replace cells that get damaged. So adult cells are those cells when the organs start development and increase in size, so these adult stem cells are actually responsible for the increased growth of the organ. And also when this organ become damaged, they repair their damaged area. Adult stem cells are said to be multipotential, which means they can only change into some cell in the body, not any cell. For example, the pluripotential can make all type of cell of the body, and multipotential they produce some type of cell, like they produce blood cell, and they are called hematopoietic stem cell, which produce only the blood cell, and skin cell, which produce epithelial cell of the skin, and also provide different type of. So this is the adult type of cell. On one hand, they produce the body tissue, on the other hand, they produce the blood cell and platelets and white blood cell. Produce red blood cell, the platelets and the white blood cell. This is the granulocyte, and also produce the monocytes. Control of growth inducer and differentiation inducer. How they are controlled, how they are usually endowment or they are non-active. In non-active form they are present, but formation of growth inducer and differentiation inducer is itself controlled by factors outside of the bone marrow. They are silent, they are non-active, but there are factors which activate them. These growth inducers or differentiation inducers, they are activated outside by factors which are present outside of the blood. For example, in case of erythrocytes, red blood cell exposure of blood to low oxygen for a long time results in growth induction, differentiation, and production of greatly increased number of erythrocytes. For the production of white blood cell, the activator factor are infections or toxic material which are present in the body. Infectious agent cause production of white blood cell. These infection agents send signals to these proteins which are called growth inducer or growth differentiation, and they are activated, and then they start the production of white blood cell. Again, it depends on the presence of infection in the body or allergic condition of the body or presence of parasite in the body. In presence of infection, the neutrophils are produced in large number. In chronic infection or allergic condition, eosinophils are produced in large condition, in allergic condition, and when there are worms, so eosinophils are produced. The tissue oxygenation is the most essential regulator of blood cell production. If any condition that causes the quantity of oxygen transport to the tissue to decrease, ordinarily increase the rate of red blood cell production. When there is less delivery of oxygen to the tissue, there is increased production of red blood cell. Also, destruction of major portion of bone marrow by any means, especially by X-ray therapy, cause hyperplasia of the remaining bone marrow, thereby attempt to supply the demand for red blood cell in the body. There are conditions like cancer when there is radiotherapy of the bone marrow, or when chemotherapeutic agents are induced, or there are some drugs when they are taken by the patient, especially chloromycin, which cause bone marrow suppression. A high altitude, it play a very vital role in the production of RBCs, because at high altitude there is insufficient oxygen. Due to this insufficiency, less oxygen is transported to the tissue and the red cell production is greatly increased. Here, for the production of red blood cell, Oxygen is an inducer factor which causes induction or production of the RBCs. A differentiation inducer is responsible for differentiations of the cells, and growth inducer is responsible for the production of the cells. So oxygen is growth inducer which causes induction and differentiation of the stem cell into red blood cell. In this case, it is not the concentration of red blood cell in blood that control red blood cell production, but the amount of oxygen transport to the tissue in the relation to tissue demand for oxygen. If a person is living at sea level, concentration of oxygen at sea level is high, and the RBCs present in the person's blood are sufficient for the transportation of enough oxygen to the tissue. But if with the same number of RBC, this person is shifted to a high altitude where the oxygen concentration is low, in that case, the RBC numbers are not that much which supply the required amount of oxygen to the tissue. So the patient easily become dyspneic at high altitude. In that condition, if a person is exposed for longer duration to low oxygen tension, this causes production of large number of RBCs, and that's why athletes are trained at high altitude, because now there are certain diseases which also cause production of red blood cell, like various diseases of circulation that cause decreased blood flow through the peripheral vessel, and particularly those that cause failure of oxygen absorption by the blood as it passes through the lung, can also increase the rate of red blood cell production. So when there is slow circulation, there is less delivery of oxygen to the tissue, there is increased production of RBCs. This is especially apparent in prolonged cardiac failure, when there is congestive cardiac failure and many lung diseases. Both these conditions, in cardiac failure and lung diseases, at one side the oxygen absorption is decreased, and on the other side there is delivery of oxygen or transportation of oxygen by the blood is decreased, because the heart pumps the blood with low velocity. So that's why the tissue oxygenation is decreased, and this decrease causes production of RBCs. RBC inducer, and this factor is called erythropoietin. A hormone erythropoietin and low oxygen level regulate the production of RBCs. When there is less RBC, erythropoietin is produced, and it causes production of RBCs. It not only increases the production of RBC, but it also decreases the maturation period of the RBC. It means that the RBCs are mature very quickly. They are produced quickly, and they are mature quickly. 90% of oxygen is produced by the kidney when both kidneys are removed, or when kidney failure is present. The person becomes anemic due to lack of erythropoietin. Production of RBCs, iron, vitamin B12 and folic acid are essential. So these vitamins and iron, they also play a major role in the maturation production of RBC. Erythropoietin is an hormone which is produced by low oxygen tension and produce red blood cell. Any factor that decreases oxygen level in the body, such as lung diseases or anemia, low number of RBC, increase the level of erythropoietin in the body. It then stimulates the production of RBC. When there is hypoxia, erythropoietin is produced, and then the production of RBCs occur. The disease causes low oxygen concentration in the blood or when there is low oxygen concentration in the environment. So disease and environment, these are the factors which are responsible for the production of erythropoietin. So low blood volume, when the blood volume is low, there is hemorrhage. It causes low blood volume, low RBC count, low hemoglobin, so it causes production of erythropoietin. Anemia means either less, obviously the volume is normal, but either the red blood cell count is low or the hemoglobin concentration in the RBC is low. So this causes production of erythropoietin. Low, when there is cardiac failure, the blood flow is slowed down and less oxygen is delivered. This causes production of erythropoietin. And pulmonary disease, low absorption of oxygen from the lung cause less oxygenation of the tissue and production of erythropoietin. And cardiac disease is hemorrhage. All these factors cause decrease in tissue oxygenation, and this tissue oxygenation actually increase production of erythropoietin by kidney. And when erythropoietin is produced, this erythropoietin acts on hematopoietic stem cell and it produces the proerythroblast, and proerythroblast is then converted into red blood cell or mature red erythrocyte or red blood cell. This is a diagrammatic presentation. When there is decreased blood oxygen, the kidney and the liver start production of erythropoietin. This erythropoietin acts on the bone marrow, on stem cells, pluripotential, hematopoietic stem cell, and they start production of red blood cell, and the deficiency is overcome.